Genome. The genes present in all the 23 sets of chromosomes in a human cell constitute human genome. Genome is responsible for the genetic properties of the organisms. Human Genomic Project is a major project for identification of all genes present in human cell and to find out their functions.
Gene sequencing, the technique of assigning positional functions of different genes of DNA molecule is termed as gene sequencing. Gene sequencing, the technique of assigning positional functions of different genes of DNA molecule is termed as gene sequencing. Electrophoresis is the process in which electricity is passed through DNA placed in agarose gel to separate the fragments having specific length and density. These fragments are termed as VNTR and can be viewed using stain ethylene bromide and observing under the UV rays.
DNA is visible under UV rays and it shows many repeating units, namely VNTRs. The variable number of tandem repeats in DNA is specific for each organism. The organism can be identified by observing the VNTR pattern. Cells adhered to clothes and other objects or bloodstain are analyzed in forensic labs in order to isolate DNA of the cells, and then the VNTR pattern of cells are found out. Matching VNTR if found, that can be traced back to the same person because no two persons have the same VNTR. Hence, VNTR pattern of DNA obtained from place of crime can be used for identifying criminals. The process of DNA fingerprinting is shown in picture. Gene modification 2. RNA interference. Gene modification can be done by silencing mRNA also. That is, certain genes are not allowed to express by interfering with mRNA so that the gene action can be modified as some needed proteins are not formed. Plants can be made resistant to a nematode, namely Neodymium incognita, by this method. The nematode cannot live in the modified plant because its essential proteins cannot be formed in that plant cells, and this technique is used for controlling the worms.
Work out the questions and try to find out the answers.